Professor Trishina, could you try your slide?
Sure. Can you see it?
Yes, that's very clear. Thank you. And Dr. Shanting, could you please share your slide?
Well, I just did that, but I can try it
Yes.
again. Sorry. Sorry, can you?
Oh, that's great.
Yeah,
Thank you.
thanks. So we have 10 minutes, don't we? For each presentation.
Yes, 10 minutes, because I mean, we
10
have, thank you. First of all, thank you, Dr. Trishina, Professor Trishina, and Dr. Shanti. This is your name, I know.
Thank you.
And thank you for joining us to this webinar. And we have 10 minutes just to show some ideas about your work, and then trying to have some time for questions to discuss the whole in the biology of mitochondria and mitochondrial dynamics specifically, and mitochondrial membrane contact sites, but also the relation to the disease and to pathophysiology mechanisms and so on. This is the point.
Thank you.
And Professor Steeman, could you please share your slide?
Yes.
Thank you so much.
Is that okay?
Yes, that's okay.
Hold on. Yeah, like this.
Thank you.
All right.
And Dr. Bikoff, could you please share your slide?
Just a moment.
Yes, thank you.
Is it okay?
Yeah, that's great. That's great. And Dr. Ziganter, could you please share your slide? Thank you.
Yes.
Hi, everybody.
Yes.
Hello, Dr. Lenner.
Muy buenas tardes.
Muy buenas
Señor
tardes.
Palau, profesor Palau.
Good afternoon and bonjour.
Okay,
Good afternoon. Buen día.
Bonjour, bonjour.
Okay, thank you so much. And Professor Palau, can we start?
Yes, we are ready. I think everybody's ready now. We can start. Yes.
Yes, okay. Every respected professors, and this is Bella, the assistant editor of Journal of Translational Genetics and Genomics, and warmly welcome to attending our webinar chaired by Professor Palau. The following is a brief welcome speech by our chair, Professor Palau. Okay, please, Professor Palau.
Thank you very much, Bella. Thank you all of you to attend the meeting. Good afternoon from Barcelona in Spain, and welcome to all of the speakers and attending academics, attending scholars. Thank you for your kind participation. I mean, it's my pleasure to lead this webinar on mitochondrial dynamics and membrane contact sites with an interest in both human physiology and biology and disease pathophysiology. And I would like to thank the Journal of Translational Genetics and Genomics for hosting it. and providing the opportunity to discuss with various topics on this important area of cell biology and a new field, I think it's a quite new field of research in human disease and pathophysiology, and of course, oriented to therapeutics or to new molecular targets or cellular targets for the helping to solve the disease in people affected by this kind of disorder that involve these mechanisms. So we are going to move to, after this presentation, to the webinar. The webinar is based in 6 professors and doctors that they know very well the field as in a specific research they are doing in their own lab. And it's my pleasure to present the first speaker, that is Professor Eugenia Trushina. She is from the Department of Neurology and Department of Molecular Pharmacology and Experimental Therapeutics at the Mayo Clinic in Rochester, in Minnesota, in the United States. And the topic she's going to address this afternoon here in Spain, I think the morning in the States and the evening in East Asia and Pacific, is therapeutic interventions to improve mitochondrial dynamics in neurodegenerative disorders. The research interest for Professor Trushina is mitochondrial dysfunction caused by environmental toxins, chemotherapies, drugs, and genetic factors in multiple neurodegenerative disorders, including Huntington's disease and Alzheimer's disease, aging, and chemotherapy-induced peripheral neuropathy. So, Professor Trushina, it's your time, and thank you for joining us to this nice webinar. So you can share your presentation with us, your PowerPoint presentation, and starting your talk.
Could you please
Yeah,
stop
thank
sharing
you.
your screen?
And please,
I cannot share when somebody else is sharing. Professor Paulo, could you please stop sharing your screen?
Okay, I was not sharing my. I'm not sharing my.
You're still sharing. I cannot
Yes.
start screen share while the other participant is sharing. So
So
you must.
maybe it's Bella, you're sharing your screen now. You should
Yes.
stop.
okay.
Okay,
Most
now
of mine was from Bella Engineer. Thank you.
Okay, now I can start. Thank you so much for your introduction, and thank you so much for inviting me. I will be very happy to talk about our work in the laboratory that I direct, where we focus on mitochondrial neurobiology and therapeutics development. So we have robust pipeline from basic science discovery through translation and application. When we're looking at mitochondria dynamics and function in respect to human diseases, Alzheimer's disease in particular. So we have multiple models, animal models, cellular models to study mitochondria function, mitochondria dynamics. We develop and adapt advanced techniques that I will share some of them with you today. And we eventually, through all this discovery, came to the development of mitochondria-targeted small molecule therapeutics that I will again share some of data with you. As you probably know, Alzheimer's disease is a devastating disorder. There is no disease-modifying cure. The development of therapeutic is hampered by incomplete understanding of disease etiology. It's not clear when the disease starts. It's not clear how it is developed. It's definitely multifactorial disorder. And it's very hard to target something in particular, as we all probably know. Targeting A-beta is controversial with not clear outcomes. So the mitochondria cascade hypothesis is a very interesting alternative suggestion for amyloid beta cascade hypothesis, where the emphasis on mitochondria is now very well documented and supported by evidence showing that mitochondria could be contributing factor to the Alzheimer's disease and underlying factor of the Alzheimer's disease. What I'm going to show you that actually mitochondria also could be the drug target for Alzheimer's disease. So why all this complexity, and how is it all possible? It all converges on incredible mitochondria origin. So being Ex-individual organism, bacteria, mitochondria maintain a lot of interesting features, including mitochondrial DNA and behavior, which is very unique to intracellular organelle. So from the very basic understanding or mechanisms that were assigned to mitochondria historically, like ATP production, apoptosis, calcium buffering, it is now clearly recognized that mitochondria play a much greater role. I listed here some of the mechanisms and pathways that mitochondria are involved, and you could see that it ranges from immediate response to any fluctuations in the energy demand, in survival of the cell. So mitochondria basically acquire the role through the necessity to survive within the nucleated cell to become like a patrol, immediate responder that communicates with the nucleus, with intracellular compartments to ensure cell survival. So mitochondria utilize a lot of their metabolites to communicate to within the cell, again, to ensure the shutdown or activation of pathways essential for energy homeostasis and cell survival, ultimately cell survival. So our therapeutic hypothesis is that we could actually harness this mitochondria signaling through triggering mitochondria tricking mitochondria into mild energetic stress conditions. So what we discovered that we could use mitochondria complex 1 as a small molecule druggable target where partial but specific inhibition with a small molecule with lower binding affinity, so it's about 30% of complex 1 inhibition, triggers the response to mild energetic stress. So this mild energetic stress is very much reminiscent of response to stress associated with mild exercise or caloric restriction. And it's actually this multifaceted response activated by mitochondria through the response to mild loss of energy production. And the outcomes now include all these mechanisms that is very hard to engage simultaneously. So we have shown now in multiple publications that this targeting of complex 1 with small molecules could improve energy homeostasis in vitro and in vivo, glucose uptake and utilization, which is the very first thing that is affected in patients with a prodromal stage of Alzheimer's disease, mild cognitive impairment. It increases the activation of resilience genes, which actually in wild type animals leads to enhanced survival, lifespan and health span, which is very important because aging is the greatest risk factor for AD. So if compounds could improve healthy aging, they could delay the onset of Alzheimer's disease. We see the reduction of A beta and phospho-tau, which is also associated with increased autophagy. And we focus, and I think that's one of the major mechanisms of neuroprotection. We improve mitochondrial dynamics and function, reducing oxidative stress and inflammation. So the compound that we use, we call it tool compound CP2 compound 2, is a small molecule tricyclic peron compound. It has very good drug properties and very safe over chronic administration through life or 14 months after the onset of Alzheimer's disease. We know now exactly where it binds to complex 1 through cryo-EM experiments with isolated ovine complex 1. So here I will talk about only one of the studies. We now conducted about 6 studies at different stages of the disease and different mouse models of Alzheimer's disease. This is a very robust animal model, APP-PS1. The mice express mutant human APP or PS1 proteins that are associated with Alzheimer's disease in humans. We treated these animals from 9 to 24 months of age chronically through drinking water with our compounds. This is the stage when animals already have the ongoing neurodegeneration and severe cognitive decline impairment. Very extensive characterization of these animals demonstrated that we have reduced A beta in the brain. We have improved LTP and restoration of dendritic spine morphology, so essential components of cognitive function and synaptic activity. In vivo, we could measure the improved glucose uptake and utilization in the brain tissue of these mice. We blocked the ongoing neurodegeneration, and we have reduced inflammation. Now I will talk in details about mitochondria dynamics and function. So mitochondria are very dynamic organelle. They feast, they fuse, they have their life cycle, and life cycle is very important because that's the renewal of organelles. So through biogenesis, there is the production of younger, healthier organelles, and through mitophagy, there is a removal of damaged organelles. When this pathway slows down with age or through the disease, the accumulation of damaged organelles, the reduced biogenesis, the production of healthy organelles, inevitably will affect energy production and bioenergetics of the cell, which is very important for such highly energy demanding cells as neurons. Here is the depiction of axonal trafficking. So it's not only biogenesis and mitophagy, it's also the motility of mitochondria along microtubule tracks in axons, because they have to get to the distal sites, and sometimes motor neurons have the axon of 1 meter in length. So if mitochondria cannot dock in the dendritic spines or cannot get to the axonal terminal, the cell will not have energy supply for synaptic function and for cellular development and connection. So it's extremely important. In aging and neurodegenerative disorders, both mitochondria biogenesis, mitophagy, and axonal trafficking are affected. In Alzheimer's disease, axonal trafficking is specifically affected through hyperphosphorylation of tau protein, phospho-tau, which is an integral component of microtubule tracks that represent the highways which mitochondria use for trafficking. So to look at the effect of our compounds on mitochondria, morphology, dynamics, and function, we adopted multiple advanced techniques. So this is classical two-dimensional electron microscopy. You're looking now at the brain tissue, hippocampal brain tissue, with hippocampal neurons aligned very nicely. You could see cell bodies on the right. So these are nuclei and the soma, and you could see the protrusions, which are axons. So we know precisely what we cut out. This is beautiful representation of hippocampal tissue. And you could see mitochondria right there, some of them in the soma, some of them along the axons. But you cannot see dendrites because usually they are perpendicular to axons. Right now, we adopted serial section phase scanning microscopy. So this is the technique when we learn now how to prepare the tissue without any artifacts. And now we have the ability to scan the tissue in very thin slices and image of the slice using electron microscopy. So now we have the tissue consecutively sliced through. And when we reconstruct these slices, we could, from the exactly same tissue, obtain now three-dimensional images of what you now could see as the hippocampal neurons. These are their axons, but we also could see very nicely dendrites. Why is it important? Because these little formations, mushroom-like formations, are dendritic spines. And I will show you what's happening with these dendritic spines in Alzheimer's disease. And compared to non-transgenic mice, actually, I can tell you right away that this is a nice mushroom-like active dendritic spine, which is mature and which is absolutely fine to participate in synaptic transmission, which underlines memory, but they are immature like that. And this is a normal process. So it's not like everything should be mature or mature. So, but if you have immature filopodia-like dendritic spines, which we see in Alzheimer's disease, these dendritic spines are not efficient in signal transduction and synaptic function. We also could use the same approach to look at mitochondria distribution in axons and dendrites, and you could appreciate right away that mitochondria are not created equally in different compartments of the cell. There are much larger mitochondria in dendrites and smaller, more motile and dynamic mitochondria in axons. So this is the close-up of the dendrites. You could appreciate the complexity of the dendritic spines. So this is a dynamic process. They go up and down, develop through life of the dendrite and the distribution of mitochondria. Please note, this is a very interesting structure that we actually identified in 2016 when we first adopted this three-dimensional reconstruction. We call this mitochondria on a string. That has never been reported before. And what it is, these are bulbous parts of mitochondria connected with the very thin tunnels. So these are just double membranes. They do not have any matrix. And you could see how thin and long this is. So if you do not apply these advanced techniques, you think that on two-dimensional EM, these will be like individually fragmented mitochondria, but it's actually one mitochondrion called MOS, mitochondria on a string. So we're very interested in these formations, and I will talk a little bit later if I have time about that. This is the reconstruction of mitochondria in the ER. So also very difficult to do in three-dimensional space, but now we have the ability to do that. Using this technique, we'll look at our mouse brain tissue. So these are non-transgenic mice. We'll look at mitochondria morphology, and you could see nice, uniformly elongated mitochondria in the mouse brain of non-transgenic mouse. We also did this mitochondria ER reconstructions. We look at the extent of ER wrapping up mitochondria and the distance between mitochondria ER, because it's very important for Merck formation. In the brain of Alzheimer's mice, you could see, as I mentioned, the formation of mouse. So a lot of mitochondria are in this stage. And in 2016, we actually characterized this in 6 mouse models of Alzheimer's disease and hypoxia. And we came to the conclusion that the formation of mouse is basically the first line of defense of mitochondria against fragmentation under conditions of stress. Because if mitochondria will be fragmented, it could be removed. If mitochondria are removed through autophagy, mitophagy, because small mitochondria could be removed and tagged if they lose membrane potential. And if biogenesis, process of biogenesis is diminished, there may be no mitochondria. So this is an interesting defense mechanism that we think mitochondria engage, waiting for conditions to normalize. And this formation of mouse actually helps to maintain residual mitochondria function and energy production. What we noticed that these mouse are very extensively wrapped with the ER, much greater than any normal mitochondria. And when we treated our mice with our compounds, we saw that mitochondria morphology resembles now more the morphology of non-transgenic mice, and mitochondria ER sites are significantly reduced. You can look at the quantification of these mitochondria ER contacts right here, non-transgenic, Alzheimer's mice, and CP2 treated mice. This is quantification of mouse, just specifically mouse. Why is it important? Mitochondria ER stress associated with extensive interaction of mitochondria and ER is implicated in multiple disorders, including Alzheimer's disease. So these contacts are very important for multiple functions. One of them is production of lipids. And in the blood of patients with Alzheimer's disease, the levels of ceramides, that's the lipids that produce specifically at mitochondria ER contacts, are elevated. Our mouse model is very translational. So our mice have exactly the same elevation of exactly the same ceramide species in blood. So we're now looking at peripheral potential blood biomarker of therapeutic efficacy for our compounds, because you could see in red that treatment with our compounds reduces the levels of ceramide in blood, which correlates with reduced mitochondria ER contacts that we can see in the brain. So we conclude that our compounds improve mitochondria ER contacts and mitochondria morphology. What about axonal trafficking? So we developed this technique a while ago. This is, you're looking at motility of individual mitochondria. So all white dots are individual mitochondria in the axon. The cell body is in the upper left corner, and the axonal terminus is on the right. So you could see that some mitochondria are completely stationary and the other move up and down. So it's anterograde or retrograde direction as we speak. This is now imaging of the same axon every second. So 600 seconds, 10 minutes. And this chymograph allows to identify the position of individual mitochondria through this 10 minutes of imaging. And you could see that these mitochondria moved non-stop without a single hesitation. These mitochondria, or actually it might be few of them, fused together, or they could be dividing. They never really moved. But here you could see that there is now definitely two mitochondria. It could be mitochondria division point. Another mitochondrion here was stationary, then it moved very rapidly, then it became stationary again, moved. So it's a very interesting way to look at what's happening with mitochondria in neurons. So these are mouse neurons from embryonic mice. And we see, for example, that in this Alzheimer's mice, inhibition of axonal trafficking occurs very early in embryonic stage, before we even see any A beta. So usually this type of inhibition is accounted for phosphatide, but multiple other mechanisms could be engaged. We developed now the ability to culture human neurons from patients who carry ApoE4 alleles. So this is the device.
1 minute more because we are a little late.
It's the last one. Yeah. So this is the same kind of thing that we'll look now at axonal trafficking in human neurons, and you could see that if we measure axonal trafficking, treatment with our compounds restore it. So this is basically the conclusion that our compounds activate multiple mechanisms of neuroprotection, including the improvement of mitochondrial biogenesis turnover and axonal trafficking. And this program was selected by first Alzheimer's Drug Discovery Foundation, Huntington Discovery Institute, and now Blueprint Neurotherapeutics Network, with the ultimate goal to produce complex 1 inhibitors for clinical trials and be in phase 1 by 2026. So thank you for your attention. And this is the teamwork, huge teamwork. I'm very grateful for all the people who were working on this project and funding agencies.
Okay, Dr. Tushina, thank you very much, because this is a very interesting talk. And we are going to have at the end of all the talk, we are going to have our own table to discuss all the points. But I mean, we can discuss with you later the approach, the therapeutic approach based in neuroprotection that has been the major topic of your talk. Thank you very much again. And we are moving to the next one. The second one is going to be given by Dr. Shanti Balasubramanian. She is the Children's Hospital at Westmead in Sydney, Australia. The topic she's going to address is CRLS1, a novel cardiolipin biosynthetic effect related to the phospholipid metabolic disorders. And the major research interest by Shanti is the lysosomal storage diseases, neurogenetics, mucopolysaccharidosis, inborn errors of metabolism in general, rare diseases in general, and medical genetics, mitochondrial diseases. Please, Dr. Balasubramanian, thank you very much for joining us, and it's your time.
Thank you. I'd like to thank the organizing committee for offering me this opportunity to share this collaborative effort, which involved several colleagues, including those in Canada, Ottawa, Canada, University of Amsterdam, and across Australia, in conclusively working up 4 patients with a novel mitochondrial nuclear gene defect affecting cardiolipin biosynthesis. So this proban, female proban, was born to non-consanguineous Lebanese couple at 34 weeks. She was small for gestational age and had non-immune hydrops fetalis. Her clinical phenotype actually evolved quite rapidly and was fatal, comprising cardiomyopathy, which was initially identified at 33 weeks, and a postnatal echo at 2 days showed a left ventricular non-compaction, which progressed quite rapidly to hypertrophic cardiomyopathy by 7 weeks of life. Clinically, she displayed central hypotonia, myoclonic jerks within the first few days of life, and declined rapidly over the next few weeks, becoming encephalopathic with bulbar dysfunction and autonomic instability. Additionally, she developed bilateral auditory neuropathy, bullseye maculopathy, and nephrogenic diabetes insipidus. The progressive neurological deterioration resulted in redirection of care towards palliation, and unfortunately, she passed away at 3.5 months. So the baseline investigations as listed, including blood lactates, were essentially quite normal. Her brain neuroimaging at 6 weeks and a repeat study at 10 weeks, including a spectroscopy, was normal. And she had an acute care trio whole exome sequencing at day 10, which was predominantly phenotypically driven, focusing on cardiomyopathy, and hydrops panel was non-contributory. So at this stage, I was consulted, and we had organized for reanalysis of the whole exome data for pathogenic variants, specifically looking at mitochondrial nuclear genes, and we identified a homozygous change in LERM4, resulting in a tyrosine to histidine substitution, which was confirmed on Sanger sequencing, and the parents were heterozygote carriers. The substituted amino acid is highly conserved across a wide range of animal species. However, it was situated upstream to the MTS, or the mitochondrial targeting sequence. Hence, it was felt that it would not probably be present in the mature form of protein and was not predicted to affect the cleavage site or cause an unfavorable polarity change that could impede mitochondrial localization. We went ahead to look for any evidence that could suggest LERM4 dysfunction. So the respiratory chain enzyme activity in skeletal muscle homogenate showed primarily complex 4 deficiency, which was in opposition to what we would expect in LERM4, where you'll get a combined complex 1, 2, and 3 deficiency because it involves ion cluster defects. So we were quite confident at this stage that LERM4 was not the candidate gene of interest, and we went into the realm of looking into novel mitochondrial nuclear genes. So further reinterrogation of the whole exome data led to the identification of a homozygous variant in CRLS1, which had no associated human phenotype at that stage. This results in an isoleucine, so non-polar change to a polar asparagine at the start of a highly conserved transmembrane domain, which is predicted to affect the stability or mitochondrial localization of CRLS1. It was also absent in NOMAD, and the in silico predictions were that it's potentially deleterious. So we had to understand what the role of CRLS1 was. So CRLS1 is involved in cardiolipin metabolism, which is a 2-step process comprising initial biosynthesis that results in the formation of an immature cardiolipin that subsequently undergoes several modifications through the activity of remodeling proteins, resulting in the formation of a mature cardiolipin. The cardiolipin synthase 1 enzyme encoded by CRLS1 has a major role in cardiolipin biosynthesis by converting phosphatidylglycerol into cardiolipin within the inner mitochondrial membrane. And mutations in tafazine, or previously known as TAZ, cause Barth syndrome, which we know is characterized by cardiomyopathy, skeletal myopathy, altered growth, and neutropenia. Now, the biological role of cardiolipin, which is also commonly known as the signature phospholipid of mitochondria, it constitutes approximately 15 to 20% of the total mitochondrial phospholipids. It's a key component of the inner mitochondrial membrane that facilitates the assembly and stability of large protein complexes, including that of the oxidative phosphorylation system and the MICOS, or mitochondrial contact site and crystal organizing system. So apart from this, it's also important in maintaining the mitochondrial crystal morphology and stability, quality control and dynamics, mitochondrial mitophagy, and apoptosis. Therefore, it's conceivable that alterations occurring within the cardiolipin metabolism may negatively impact the activity of a variety of mitochondrial proteins and enzymes, including that of the oxidative phosphorylation complexes, thus compromising mitochondrial function and dynamics. So to investigate how CRLS1 contributes to disease pathogenesis, mitochondrial respiration and OXPHOS protein stability were measured in patient fibroblasts and healthy control. High-resolution respirometry showed a 20% reduction in complex 4 activity in the ATP generating or phosphorylating state 3 and non-ATP generating or non-phosphorylating state 4 condition, while, so this is in patient fibroblasts, while both state 3 and 4 respiration of complex 1 were comparable. So hence again, excluding a possible differential of LERM4, where you would expect complex 1 to be affected. Next, reduced mitochondrial protein synthesis was determined by the decreased de novo mitochondrial translation rates for all 13 mitochondrially encoded proteins in the patient fibroblasts, despite a 2-fold increase in mitochondrial DNA copy number. And this increase is thought to be due to be a compensatory response to the reduced mitochondrial protein synthesis that is attributable to cardiolipin loss, as it facilitates mitoribosome association in the inner mitochondrial membrane. These defects and compensatory response of the mitochondrial gene expression machinery was closely resemble what was described by the senior author in Professor Alexandra Filipovska in CRLS1 mutant knockout cell lines. So again, you get reduced mitochondrial protein synthesis and 3 different forms, so minor, moderate, and severe CRLS1 knockout models, despite the increases in the mitochondrial DNA copy number. So we know that cardiolipin is important for maintaining the mitochondrial network integrity. So MitoTracker Orange was used to examine the mitochondrial morphology in patient and control fibroblasts. And here we see a very fragmented mitochondrial network towards the periphery of the cells, but towards the nucleus, they have retained their structure. And with rescue experiments, we are able to demonstrate that the reticular morphology is restored with long tubular structures distributing throughout the cell. However, with rescue in LERM4, the mitochondrial integrity is not being retained. Now, crystalline morphology examined under transmission electron microscopy was highly disordered with smaller, rounder, and contracted mitochondria in patient cells compared to control mitochondria, indicating impaired crystalline folding, which have previously also been reported in Barth syndrome patient cells. So together, these morphological defects and rescue experiments indicate that the I109 variant impairs cardiolipin-associated regulation of mitochondrial membranes, likely resulting in the OXPHOS defect observed. Next, we looked at cardiolipin abundance as well as a cell chain composition profile of both patient and control fibroblasts, which were examined under nanoelectrospray ionization accurate mass spectrometry. So there was a significant reduction in total cell cardiolipin in the patient fibroblasts, and the cardiolipin SL chain composition in fibroblasts showed an enrichment of larger SL chain containing species with a depletion of the shorter chain species. So this data functionally validate once again the pathogenicity of the I109 variant, illustrating that it causes alterations in cardiolipin levels and SL chain composition. Subsequently, we were able to identify other patients, 3 additional patients, 2 of whom were from Sydney and who remain a diagnostic odyssey for several years. So this family also came from the same ethnic background, but they are unrelated and they have very similar clinical presentations, and they were identified to have the same homozygous I109 variant. A 4th patient was identified by Professor Kim Boycott and Dr. Michael Garati in Ottawa, Canada. So this is what appears to be a milder phenotype, so a 19-year-old male with permanent disability and poor functional status, first presented at 3 months with slowly progressive encephalopathy and global developmental delay, sensorineural hearing loss, congenital nystagmus, and decreased vision. So he had a compound heterozygous variant identified in CRLS1, but the paternally inherited variant was thought to be a milder variant because of the conversion of the proteins was not thought to affect the CRLS1 protein as severely as the I109 variant. So this was consistent with the clinical presentation that this patient had a milder phenotype as opposed to the first 3 patients with the homozygous ones I109 that were all fatal within the infantile period. And cardiolipin abundance here showed a trend towards reduced cardiolipin in this patient compared, which is milder in comparison to the patient with the I109 variant. So in conclusion, combining multi-omics technologies with clinical phenotyping in these patients has demonstrated that pathogenic variants in CRLS1 cause a novel mitochondrial disorder with a disease spectrum ranging from premature death in 3 infants to permanent disability in early adulthood. And the characterization of these novel CRLS1 variants advances our understanding of the essential role of cardiolipin in human disease. Thank you, and I'd like to acknowledge all the people involved in this work. Thanks.
Thank you very much, Shanti. I mean, this has been a nice presentation talking about a new disease related to mitochondrial metabolism, specifically phospholipid metabolism. We are going to talk later on about that because it's, I mean, I know that you have just published in Human Molecular Genetics the paper in the last month, I think, in October.
Yeah. Yeah.
Yes, okay. Congratulations. And it's really very, very interesting because it's increasing the number of cases and the number of diseases related to the phospholipid metabolism and cardiolipin specifically. Thank you very much. We are moving now to the next presentation by Professor Louis Renner. Professor Renner is the Mitochondrial Medicine Research Center in Angers University in France, Angers. And he's going to talk about mitochondrial dynamics in the degenerescence of the optic nerve. I mean, his interest in mitochondrial DNA, mitochondrial diseases, and phosphorylation, and also genomics, human genetics, and next generation sequencing, the Centre Hospitalaire d'Angers. the Centre Universitaire Hospitalaire d'Angers in France. Please, Guillener, Professor Guillener, you are waiting.
Okay, do you hear me?
Yes,
perfect. You hear me? Do you have my PowerPoint in front of you? Yes?
Yes, we have.
Okay, so I start. So I'm also very pleased to have the opportunity to present some of the data that we are generating in my lab, which are both oriented on preclinical and clinical studies on inherited optic neuropathies. So you all know that dominant optic atrophy is a mitochondrial disease leading to blindness. So it involves disruption of mitochondrial network, which ultimately leads to, let me see if I can see, okay, which leads to the degeneration of the optic nerve, meaning that it's not a perception of light which is affected, but a transmission of the visual information from the retina to the brain, which is affected. And this is leading in most cases to a central scotoma in patients and the preservation of the peripheral visual field. So we are facing a mitochondrial disease, and obviously when we address the reason why the optic nerve is degenerating, we have to understand first what is the mitochondrial defect associated to mutation responsible for this disease. And the second question that we have to address is the clinical specificity, meaning why only the optic nerve is degenerating, whereas all the other tissues are preserved. So, as you might know, we are involved in a molecular diagnosis of inherited optic neuropathy in France. So it means that we gathered all the DNA samples from all the patients in France, and we have published recently overview of the molecular data, molecular diagnosis on 1,680 patients with inherited optic neuropathy involving dominant optic atrophy, recessive and Leber hereditary optic neuropathy. And we have now a panel including 88 genes, and you have here on the left the most frequently genes that have been identified, mutated, and on the right the more rare genes that are identified. And you see here on the left that the main genes obviously responsible for the degenerescence of the optic nerve is OPA1. And in green, you have additional genes that are involved in mitochondrial dynamics. On the right, concerning the more rare genes that we identified in optic atrophy, you have 4 genes that are also involved in mitochondrial dynamics. And importantly, what we figure out is that all these genes that are frequently involved in dominant optic atrophy are related to either mitochondrial fusion or mitochondrial fission. Supporting mitochondrial fusion, we identify mutation OPA1, MFN2, SPG7, AFG3L2, and supporting mitochondrial fission, we found mutation DNM1L, OPA3, and MIEF1. So this means that there is a critical requirement for mitochondrial dynamics to maintain the physiology of the optic nerve. And the question is, why the optic nerve? And now we have to enter in the second question of my talk. It's why, when mitochondrial dynamics is affected, you have a degeneration of the optic nerve. And one peculiarity of the optic nerve, it's the fact that it's facing 2 different physiological conditions. When you see the retinal ganglion cells that are forming the optic nerve, their axon is not myelinated inside the eye, just to prevent the blurring of the transmission of the visual information through the retina. Okay, but once the axon is passing the lamina cribosa, which is a physical membrane closing the eye in between the eye and the optic nerve, axons are getting myelinated. Okay, and this is true until the synapses of the cell. So we have been looking for the distribution and the structure of the mitochondrial network in both parts of the axons of retinal ganglion cells. And this is what we found out. So here on the left, you have a whole retina from a mouse model. Okay, in green, you have the distribution of mitochondria. And you can see that mitochondria are pretty abundant all along the different layers in the retina until the entry in the optic nerve. Once we cross the lamina cribosa here on this part of the immunofluorescence, and we label for mitochondria and MBP, which is a specific protein of the myelin part of the axon, you can see that where you have a lot of myelin, you have very few mitochondria. Where you have no myelin, you have a lot of mitochondria. And this stays through all along the myelinated axons, where you can see that the abundancy of mitochondria is pretty low at the chiasma, is pretty low in the optic tract, and until the synapses here on this side, you still see that the abundancy of mitochondria is pretty low. So we further look by electron microscopy for the structure of the mitochondria upstream, before of the lamina cribosa and after the lamina cribosa. And you can see that here on the left, where axons are not myelinated, you have huge abundancy of mitochondria, and they are round with a very clear matricial content. Oppositely, if you go in the myelinated part of the optic nerve, you can see here the myelin very easily, and there are less mitochondria, the abundance is much scarcer, and mitochondria are elongated with a dark matricial content. And we got this exceptional picture on the lamina cribosa, where you can see the transformation of this type of mitochondria in this type of mitochondria here associated to the myelination process of the axon of the retinal ganglion cells. So what we believe is that we really need all the actors of mitochondrial dynamics for the anterograde transport of mitochondria to transform this type of mitochondria in this type of mitochondria. And we also need the actors of fission to transport back, retrograde transport of mitochondria from the synapses to the soma. And obviously we need DRP1, OP3, and MIF1 for this activity. So if I come back to my scheme, in the unmyelinated part of the retinal ganglion cells, you need a huge amount of mitochondria to support the transmission of action potential upstream of the lamina cribosa. Once you pass the lamina cribosa, retinal ganglion cells axons are getting myelinated. You have less amount of mitochondria, and they are more elongated and with a darker matricial content. So mitochondrial dynamics is crucial for the retinal ganglion cell physiology. And to conclude this talk, what we know now is that so on the left side, you have a wild type condition. On the right side, you have the condition of a patient with a dominant optic atrophy, and this pallor, which is specific of the dominant optic atrophy. If you have either overfusion or overfission, both conditions are leading to dominant optic atrophy. So DOA is mainly related to mitochondrial dynamic defect. Both excess of, so equilibrium between fusion and fission is crucial for the optic nerve physiology. And the last point is that the therapeutic window between excess of fusion and excess of fission is very narrow, and therefore a challenge for our future therapies. And I just want to end here, thanking the people that contributed to this work, the patient association that also contributed to support financially this work, and obviously the university and University Hospital of Angers that are supporting our laboratory. And I thank you for your attention.
Thank you very much, Professor Leonard. Really impressive, the difference between the morphology, and I understand the physiology of mitochondria in both sides of the cribosal lamina cribosa is really impressive. And we have the opportunity to discuss a little later on in the round table. But really, congratulations for your work and a very nice talk and very nice, interesting findings. Thank you very much again. And we're moving to the next talk that is going to be given by our next speaker, that is Dr. Yuri Bykov. He's at the Department of Molecular Genetics in Rehovot in Israel at the Weizmann Institute of Science. And the topic of his talk is going to be mitochondrial ribosomal proteins use unconventional import signals. I mean, his research interest is related to electron microscopy imaging processing, and also targeting and translocation in mitochondrial proteins. Dr. Bykov, please.
Hello, thank you very much for such a nice introduction, and thank you for your invitation to talk at this webinar. So now I'm going to focus on this project that concerns mitochondrial biogenesis. Mitochondria has their own genome that encodes 8 genes in yeast and 13 in humans, and their own gene expression machinery, but the majority of mitochondrial proteins, which are around 1,000, are encoded in the nucleus and are expressed on cytosolic ribosomes, and then exist in the form of unfolded precursor proteins that get targeted and translocated to the mitochondria. And this process needs to be very precise because unfolded precursors are toxic to the cell, so the cells need to get rid of them quickly. And there is large diversity of precursor proteins because they need to be imported into different subcompartments of mitochondria, like outer membrane, intermembrane, space, intermembrane, and matrix. And all of these proteins have specific targeting signals that are recognized by mitochondrial import machinery. And in fact, we don't know the diversity of these signals so well because only few model precursor proteins were studied in vitro. In this work, we focus on proteins that go to the matrix. And for these proteins, the most canonical signal is so-called matrix targeting signal, MTS, or pre-sequence. It's an N-terminal peptide that is recognized by receptors on the outer membrane of the mitochondria, receptors TOM20 and 22 of the translocase of the outer membrane, or TOM complex. Then it gets translocated through the TOM40 subunit and handed over to the TIM23 complex, which is translocase of the inner membrane and imported on membrane potential dependent manner. In the matrix, MTS is cleaved off and its usual length is around 30 amino acids. However, it was noticed several years ago that there is a class of proteins that go to the matrix but do not have a conventional cleavable MTS. Interestingly, a lot of such proteins are components of the mitochondrial ribosome, a very spectacular molecular machine. And the yeast one, and we work on yeast, Saccharomyces cerevisiae, has 80 proteins that are called MRPs, compared to the bacterial homologous ribosome that has only 55. And in this project, we were interested about these MRPs that do not have a canonical targeting signal compared to normal matrix proteins with a cleavable MTS. So which signals do they use? Which import pathways do they use? And why do so many MRPs do not have an MTS? So to address the signal question, first I collected all available information about N-termini processing of the MRPs. This can be found in original papers, N-terminal proteomics data, predictions on UniProt and special software, and actually a structure itself can also be informative. Finally, we concluded that around 25% of MRPs do not have a canonical cleavable MTS, and we selected 15 proteins for further functional study. And in this functional assay, we ask a very simple question. Okay, the N-terminus doesn't have a cleavable MTS, but maybe N-terminus still contains the signal. It's just the targeting signal. It's just not cleaved off. And to answer this question, we define N-terminus as 30 amino acids, like a cleavable MTS, and then decided to check by either deleting this N-terminus or using its N-terminus fuse to GFP, whether the N-terminus can act as a targeting signal. And if it can, we expect that N-terminus on itself will target the protein GFP to mitochondria as well as the full length, while the deletion of the N-terminus will lead to disruption of targeting. And indeed, some proteins followed this pattern. There were roughly a third of the proteins. Then third of the proteins had an opposite phenotype. The deletion of the N-terminus had no effect on mitochondrial targeting, while N-terminus on itself could not target GFP to mitochondria. Finally, 30% of proteins had some kind of intermediate phenotype. We decided to focus on this protein called MRP17, which is very conserved and doesn't have any N-terminal targeting signal whatsoever. So in the meantime, we conclude that many MRPs have a variety of non-cleavable targeting signals, both on the N-terminus and inside of the proteins. And now we move to MRP17 and use a similar assay to find out more precisely where does its targeting signal reside. And we take parts of MRP17 and fuse it to GFP and compare the signal to mitochondrial marker. And we can see that smaller regions of MRP17 in general either cannot target to mitochondria or target very weakly. And we, using these assays and also in vitro import assays into isolated mitochondria, define this region as amino acid 30 to 60, which is where the targeted signal resides. You can appreciate here that in this construct, there is still some mitochondrial signal visible in the GFP channel, while here it's purely cytosolic. So we conclude that MRP17 has non-canonical internal targeting signal. So how does it get recognized? Is it similar to regular MTS? And to answer this, we used in vitro import assay in which we use mitochondria either from wild-type yeast or the yeast where TOM20 receptor was deleted. And indeed, MRP17 was imported much less into such delta TOM20 mitochondria, meaning that TOM20 might be involved in the signal recognition. To find out if this effect of deletion is direct, we looked at the sequence of MRP17, and you can notice that the protein is positively charged. Here we show positively charged amino acids in blue. And in this region where we have identified the signal, it contains two TOM20 binding motifs that were previously characterized using NMR studies. And if we mutate these motifs, such a mutant is not imported into isolated mitochondria compared to control protein, which can be judged by the absence of the protein in these three lanes. So we also found out that positive charges both inside the signal and around it are important for protein import. So we conclude that MRP17 import depends on TOM20, but also it depends on TIM1723 in the inner membrane, which I cannot have time to show now. So overall, its recognition looks like recognition of a conventional MTS. And finally, we tried to answer the question why so many specifically MRPs do not have an MTS. And at least for MRP17, we think that explanation is evolutionary. If we look at the MRP17 homolog called S6, in the bacterial ribosome, it sits on this conserved rRNA helix, and its N-terminus is mostly free and exposed to the solvent, while MRP17 sits on exactly the same helix, but its N-terminus is completely buried in the protein-protein interaction interface. So what if this interface is so conserved that there is absolutely no possibility to change a single amino acid in the N-terminus to allow adding a cleavage site and extend the protein with a cleavable MTS? To check if this is possible in principle, we decided to add an artificial MTS and look at the function of such MRP17. For this, we created delta MRP17 mutant, which has this not very strong growth effect on respiratory media containing glycerol. And MRP17 itself can compensate for this defect, fully restoring the growth. And if we use this artificial MRP17 with additional cleavable MTS, we can see that it can restore the growth as well, meaning that adjusting the N-terminus is not so critical for the function of this protein. But we still think that evolution can be important for the development of internal signal. If you imagine a bacterial protein that needs to become mitochondrial and develop a cleavable signal, there is still in the process of evolution must be some intermediate where the cleavage site position is not optimal, like we already made it artificially. So this intermediate will be detrimental for mitoribosome assembly. And in case of positively charged RNA binding protein, which MRP17 is, it's just easier to add more positive charges and TOM20 binding motif, making it recognized similar to a regular MTS by the import machinery. With this, I'd like to conclude and note that this work was recently published. So we think that at least for MRP17, its unusual signal properties can be explained by ribosome assembly. With this, I'd like to finish and thank our collaborators in this project, Professor Johannes Hermann from Technical University of Kaiserslautern and his student Tamara Flor, who did all the in vitro experiment, and the Schuldiner lab for their help and support, and of course you for your attention. Thank you.
Thank you very much, Dr. Bikoff. I mean, really very interesting, this biochemical and basic science approach to this kind of proteins related to ribosome proteins, mitoribosomes proteins, and also to learn how it's necessary to find or to have this kind of mitochondrial signals and location signals to get inside the different compartments into the mitochondria. We are going to discuss also later on about that. Thank you very much. And now we are moving to the next talk that is going to give by Professor Thomas Simen from Canada, from Alberta, Edmonton in Canada. He's at the Department of Cell Biology in the Faculty of Medicine and Dentistry at the University of Alberta, as I said before. And the topic and the talk is going to be related to the redox control in the plasmid reticulum mitochondrial signaling. I mean, the research interest by Professor Simen is the membrane contact site between the plasmid reticulum and mitochondria as determinants in cancer and in neurodegeneration. Thank you very much, Thomas, for joining us today, and it's your time.
Okay, good morning, everyone, or good evening or good afternoon, depending on where you are. Can you hear me, and is the slide showing?
Yes, we can. Yes, perfectly.
Okay, very good. All right, so my lab focuses on the role of membrane contact sites and on mitochondrial dynamics and their roles in disease. And yeah, okay. All right. So here you can see an overview of the so-called mitochondria-associated membranes, which is a structure inside cells that had been discovered at my university biochemically about 30 years ago by Jean Vance. This structure wraps around mitochondria and controls their functions to a very large extent through the exchange of phospholipids, calcium, and reactive oxygen species that migrate between the two organelles and overall control the bioenergetics of mitochondria and the ER. So while this role is well known in the case of mitochondria, it is not so well known in the case of the ER, and that is a story that I'm going to tell you today in an abbreviated way. So, like I said before, we focus on cell biological pathways and their identification that control this metabolic crosstalk between the ER and the mitochondria. But before I get into this, I would like to list very briefly a few of the disease phenotypes that result if this machinery does not work properly. And the first example I'm going to give is the so-called PAX2 syndrome, which is based on a mutation of a protein that I had discovered in 2005 as the first protein that is responsible for connecting mitochondria to the ER. In this phenotype, as you can see in the pictures, microcephaly as well as epilepsy results in children. And this phenotype resembles actually a cluster of diseases which have all very similar phenotypes in the patients. And when you look a little bit more carefully about what proteins are affected by this type of diseases, you notice that most of the genes involved are controlling protein folding inside the endoplasmic reticulum, such as this example, which I show down here, called IER3IP1, which gives rise to a disease called MEDS, where you have dysregulation of ER mitochondrial signaling, dysregulation of the endoplasmic reticulum unfolded protein response that comes with mitochondrial dysfunction as well as ER stress. Another type of these diseases is represented by Walcott-Rallison syndrome, a case of a disease which most well-known gives rise to a diabetic phenotype in children. This disease is based on the unfolded protein response kinase PERK. And at this point, you might wonder how the control of the unfolded protein response inside the ER could give rise to defects in mitochondria. And that is what I'm going to explain in the remaining slides. So to summarize this first brief introduction, mutations in the kinase PERK that controls the unfolded protein response inside the ER give rise to Walcott-Rallison syndrome, a childhood diabetic disease that partially overlaps with MEDS as well as the PAX2 syndromes. And what these diseases have in common, in addition to a diabetic phenotype, is that there are defects in the central nervous system characterized by microcephaly as well as epilepsy. In all of these diseases, redox as well as organellar bioenergetics involving the ER and mitochondria are out of balance, leading to the defects that then give rise to the diseases. So now at this point, of course, we would like to know what the cell biological mechanistic basis is for these diseases and how these very diverse genes can give rise to such similar phenotypes. And the basic science behind these observations comes from our studies that have demonstrated how ER folding chaperones control calcium exchange between the mitochondria and the ER. Under homeostatic conditions, as shown here on the right, SERCA and IP3 receptors exchange calcium with the mitochondrion, which inside the mitochondrion is required for the progression of the TCA cycle as well as the production of ATP. This ATP production is in equilibrium with ATP production made by glycolysis. The ER chaperones that control this mechanism, we have shown to control the activity of pumping of calcium into the ER by altering the activity of SERCA calcium pumps. This is done, for example, by calnexin, as well as the protein TMX1, which can either activate or inactivate SERCA pumps. And this enzymatic function requires also the production of reactive oxygen species by NOX4, an ER protein that makes reactive oxygen species inside the ER lumen. So through this mechanism, you can see how the conditions within the lumen of the ER control what happens in the mitochondria through this calcium-mediated crosstalk. But these enzymes function under homeostatic conditions. Of course, cells can undergo stressed conditions, and under those conditions, we have also shown that these enzymes do not play a role anymore for the crosstalk mediated by calcium between the two organelles, which again uses the same calcium handling proteins, but is now boosted. So the question was, how would the calcium crosstalk be controlled under these conditions? And in work that I'm going to show you now, we found that this depends on unfolded protein response transmembrane proteins of the endoplasmic reticulum, IRE1 and PERK, which connect the two organelles and whose interaction increases with the mitochondria under stressed conditions when you have increased protein folding defects inside the endoplasmic reticulum that then boost this calcium exchange between the two organelles. And it is under conditions when this crosstalk mediated by the UPR proteins is not working properly that you get disease. The work started with a mass spectrometric analysis of the interactors of the kinase PERK by my former postdoc Arthur Bassot, who found that under conditions of ER stress, PERK interacts much more with an ER reactive oxygen producer called ERO1. He also showed that this by co-immunoprecipitation, where you can see that under ER stress, triggered by tunicamycin, but the same can be observed with other ER stressors such as tapsigargin, ERO1 interacts better with the kinase PERK. The question is, what is the significance of this interaction? Before I get into this, I just wanted to briefly mention that NOX4, as I've shown on the previous slide, that mediates the basic interaction between the two organelles, is not involved in this mechanism. So Arthur went on to characterize the consequence of the interaction of this ROS producer ERO1 with PERK under conditions of ER stress. And he first showed that this leads to oligomerization of PERK on mitochondria-associated membranes, which he detected here on a non-reducing gel. This effect is disrupted if ERO1 activity is inhibited. At the same time, he also showed that IP3 receptors undergo oxidation in an ERO1 dependent manner. This was done by a BM labeling assay where IP3 receptor that is fully reduced shows up as labeling, and it does not happen if it is fully oxidized. As you can see here, under control conditions, IP3 receptors are partially oxidized and their oxidation increases under conditions of ER stress in an ERO1 dependent manner because the inhibitor of ERO1, EN460, was able to abolish this oxidation of IP3 receptors. Now, if IP3 receptors become oxidized, this is known to activate calcium flux. So the expectation was that under these conditions, the interaction between the ER and the mitochondria would increase and calcium flux would increase as well. Now, while I don't have time to show you the calcium flux data, I'm going to show you here data where he showed that under conditions of ER stress with tunicamycin, the contacts between the two organelles increase, as shown here with a fluorescent indicator called SPLIX, where you can see the dots of contacts between the two conditions increase in wild-type cells, while this increase is not observed in PERK knockout cells or in the presence of the ERO1 EN460 inhibitor. As a consequence, in wild-type cells, the mitochondrial membrane potential goes up under ER stress, which does not happen if PERK is absent. And the same increase is also not observed in the presence of the EN460 the inhibitor of the ERA1 ROS production inside the endoplasmic reticulum. Now, for mitochondrial bioenergetics, Arthur showed that under ER stress, bioenergetics go up inside the mitochondria. They make more ATP. This doesn't happen if PERK is absent, as shown here with the red columns, and it also does not happen if ERA1 is inhibited, regardless of whether PERK is present or not. So the last question that I'm going to answer in this context is whether this increase of mitochondrial bioenergetics has a consequence for the folding environment inside the endoplasmic reticulum by measuring ER bioenergetics upon ER stress. As we can see here in the top graph with the black curve, normally upon induction of an ER stress at a very short time point, ER bioenergetics are maintained, but in the absence of PERK, or shown down here in red, or upon inhibition of ERA1, you can see that this leads to a rapid drop of the presence of ATP within the endoplasmic reticulum. ATP, of course, being necessary to eliminate any unfolded proteins that would accumulate under these conditions. So in other words, if mitochondria are not able to ramp up their ATP production upon an ER stress, then you end up getting an energy crisis within the endoplasmic reticulum within as little as 5 to 10 minutes, as shown here in these graphs. Now, we then moved on to investigate the requirements within PERK for this interaction, and we noticed that PERK contains a multitude of cysteines which are conserved and which have also been shown in the ox mouse database as undergoing oxidation during aging, as indicated here by these asterisks. And Arthur found that within the PERK luminal domain, there is cysteine 216, which is required for the interaction between the two proteins and for all the effects that I've shown to you before. And interestingly, this cysteine is close to known Wolcott-Rallison dysplasia mutations, such as this one here, which introduces a new cysteine very close to the critical cysteine for the interaction. Arthur also found that cytoplasmic cysteines are playing a helper role for this mechanism, and one of these is known to also be involved in Wolcott-Rallison syndrome mutations. So to summarize, here's the mechanism that we have described, which we predict is contributing to the Wolcott-Rallison syndrome. Under homeostatic conditions, calcium signaling maintains bioenergetics within the mitochondria as well as the ER. You have a baseline production of reactive oxygen species by NOx4. PERK is inactive. Under ER stress conditions, the alternative ROS producer, ERA1, of the endoplasmic reticulum covalently interacts with PERK, which undergoes oligomerization, which then leads to the oxidation of IP3 receptors and a boost of calcium signaling towards the mitochondria, which then turns on ATP production. And this increased ATP production is required to maintain ER bioenergetics, which then here in my summary can give rise to diseases of the mitochondria-associated membrane, one representative of which is the Wolcott-Rallison syndrome, where mitochondria are defective because this communication of ER stress towards the mitochondria is compromised. And on my last slide, I'm thanking my university as well as the funding agencies and the people in my lab who contributed to this work. Most, of course, Arthur Bassot, who was the responsible person for this project, as well as our collaborators with whom we work on additional diseases of this structure.
Thank you very much, Professor Simen. Thomas, I mean, really very interesting to see how the field of a membrane-associated, mitochondrial membrane-associated contact sites is increasing the number of diseases and the pathophysiology associated with this important microdomain between the endoplasmic reticulum and the mitochondria. Really very interesting. Thank you very much. We're now moving to the next and the last, but not least talk this afternoon here in Spain, morning, evening, is going to be by Elma Zaganjo. She's at the Department of Molecular Physiology and Biophysics in Vanderbilt University in Nashville in the United States. And the topics she's going to give us is the mitochondrial dynamics and metabolisms drive adipogenesis. So her major interest in science, biomedical science, is the role of mitochondrial function in cell fate decisions and the implication of this regulation in physiology, and today in adipogenesis. Ilma, thank you.
Thank you for the kind introduction. Can you all hear me and see my screen?
Yes, perfect. No problem.
Okay, thanks. This was great. Thanks for the invitation. I've learned a lot today. So this is kind of where my lab is moving into, but I'll today show you some of the recently published work. So my lab is interested in the role of mitochondrial dynamics and metabolism in cell fate decisions. But today we'll show you how these dynamics and metabolism actually drive adipogenesis, which is a process of formation of new adipocytes. So as you know, mitochondria are central to cell fate decisions. So whether the cells choose to proliferate and maintain their stemness, or whether they differentiate into new cell types, this is really guided by mitochondrial dynamics. So to maintain stemness, the mitochondria should be fragmented and not perform a lot of oxphos. So these cells derive a lot of their ATP from glycolysis. Now, once you expand the mitochondria through biogenesis and the mitochondria become more fused and increase their complexity, these cells can really then differentiate. Now, this is not always the case. There are some exceptions to this rule, but essentially it is thought that these mitochondrial changes in dynamics do really regulate cell fate decisions. Now, as you can see in the case here of differentiated cells, they have many fuel choices. They can oxidize glucose, fatty acids, or even amino acids to produce ATP or other intermediates for biological functions. But does it actually matter which fuel choices are taken by cells? Does that impact cellular function? And so if you look at mesenchymal stem cells that divide into this progenitor pool, that can then become numerous different cell types, such as chondrocytes, osteoblasts, myocytes, or adipocytes, for example, doesn't matter which fuels they're oxidizing. It turns out there's evidence that it does matter. So much like these transcriptional regulators, changing fuel choices also impacts their differentiation potential. So Carnell Lab at UT Southwestern has some time ago demonstrated that if you block glutamine oxidation, now the progenitor cells are less likely to become osteoblasts. Their capacity is diminished. However, there is increase for adipogenic cell fate. So we found this point very interesting, and we were thinking, if these progenitors are not oxidizing glutamine, what fuel choice are they actually taking up? Obviously, this is a critical cell type that regulates energy balance in an organism. So we wanted to study this question. So what are the metabolic pathways that drive this differentiation into adipocytes? And to study this, we use a lot of in vitro cell models. So there's a 3T3L1, which is a commonly used cell type since the 70s, but we also take these primary stromal vascular fraction cells from neonatal mice, and we can then differentiate them. These are established protocols. We use isobutyl methylxanthine, insulin, and dexamethasone to stimulate transcription and signaling and transform these pre-adipocytes into mature adipocytes that can now store lipids. And on the right here, you can see that our system is working as expected. So the undifferentiated cells pretty much do not have any lipid, whereas 6-day differentiated cells, based on these stains, oil red O and BODIPI, the stain neutral lipids, are fully differentiated. Now, we didn't want to compare metabolic changes between undifferentiated and fully differentiated cells. We thought if a metabolic pathway is driving the process, it will be activated early. So for this reason, we used undifferentiated cells and compared them to 2-day differentiated cells. These cells do not contain lipid droplets, and the transcriptional regulation is just beginning. So we isolated metabolites from the media and from the actual cells. And so what we can learn out of these metabolomic studies is how are metabolites secreted or taken up based on metabolomics from the media, but also what's remaining in the cells. So how are they consuming these nutrients? And so summarizing all this data, we find that within 2 days of differentiation, we see these global changes in amino acids. And so this is really kind of unexpected that we see such huge changes prior to this complete change in cell state. And particularly what we found interesting was that branched chain amino acids were some of the most impactful signatures that were altered. And the reason this was significant to us is because branched chain amino acids have been noted to be altered in the plasma of diabetic patients for many decades now. So we thought this regulation might actually have some implication in pathology as well. So what are branched chain amino acids? So leucine, isoleucine, and valine. In the cell, obviously, they can be used for protein synthesis, but in the mitochondria, they can be oxidized. And once oxidized, they can produce acetyl-CoA or succinyl-CoA, which can then be integrated in the TCA cycle, generating NADH and FADH2, which then drives ATP synthesis. But how can we actually study if this is at all altered during these early stages of adipogenic differentiation? So we decided to focus on leucine. And so obviously we could look at transcriptional and protein regulation of enzymes, and we did all that. This is all published. But we can also perform some functional studies. So we can use heavy labeled C13 labeled leucine on all carbons, and we can trace it. So if there is increase in oxidation, we would expect to see incorporation of the carbon from leucine into TCA cycle intermediates and subsequently into amino acids that can be generated out of those intermediates. And so we did that experiment. So if I point you here to undifferentiated versus 2, 4, 6, or 8-day differentiated cells, you will note that we see increase in percent labeling from this carbon as we induce differentiation, suggesting that induction of differentiation stimulates leucine catabolism. We've also done this through different assays. So the other thing we can do is we can take intermediates from leucine catabolism, such as isoketocorporic acid, and we can measure how much that stimulates mitochondrial respiration using Seahorse assay, for example. And by that measure, we also see increase in leucine oxidation once you induce adipogenesis. Okay, so this process is induced, but does it actually matter? Does it subsequently regulate adipogenic capacity? And so we perform that assay as well. So one thing we can do is we can knock out enzymes in this pathway and see if we actually disrupt adipogenesis. And so we decided to knock out methylcrotenyl-CoA, one of the key enzymes, and then we can measure lipid accumulation in the cell. So we can stain with oil red O, and that can as well be quantified. So we can extract the dye and see how much that's altered by knockdown of MCC1. And so we knocked down MCC1 with 3 different shRNAs, and we find that there's decrease in oil red O, suggesting that blocking this pathway also blocks adipogenesis. But how is this actually regulated? So we turned to the literature on this one, and we found that there are actually some studies where they find that master regulator of adipogenesis, the transcriptional regulator PPAR gamma, if you knock this out in mature adipocytes using a adiponectin-driven Cree model, you will see decrease in enzymes of branched-chain amino acid catabolism, and you will also see increase in branched-chain amino acids in the plasma, suggesting that branched-chain amino acids are not being oxidized. But does this actually work in our system? So to test this, we use multiple different inhibitors. We also use different shRNAs to knock out PPAR gamma in our cells. And so I will just summarize the data. So essentially, we validated the literature. So in mature adipocytes, if you knock out PPAR gamma, you affect transcriptional regulation of enzymes in branched-chain amino acid catabolism. And you can also do functional studies, and you'll find that without PPAR gamma, there is also less branched-chain amino acid catabolism. But this is only in mature adipocytes. If you look at early regulation, so when we first induce differentiation at those early time points, 1 day, 2 days, for one, there is no induction or transcription of branched-chain amino acid enzymes. So PPAR gamma is not involved in these early time points. So we know that this pathway is induced. So what's actually regulating it? So again, we looked at what's actually known about this regulation of branched-chain amino acid catabolism that's not transcriptional regulation. And so we found numerous studies where they identified SIRT4, a mitochondrial deacylase. So this is one of the key post-translational regulator of proteins in the mitochondria. Its enzymatic activity is not fully elucidated, but there's a lot of evidence that SIRT4 binds to this enzyme MCC1 that I already mentioned, and in the liver at least modulates its activity. And there are a few more proteomic studies where it's been confirmed that SIRT4 does interact with this enzyme. So that's great. It interacts in the liver and has some function, but in our system, is there actually some functional consequence and interaction between SIRT4 and enzymes in branched-chain amino acid catabolism? So I will just show you one piece of data where we knock down SIRT4. And again, we performed this functional assay where we can trace carbon from leucine into TCA cycle intermediates and beyond. And we find that even in undifferentiated cells, loss of SIRT4 impacts leucine oxidation. And moreover, we also find that in adipocytes, as soon as you induce differentiation within 1 day, MCC1 activity goes up, but also it's regulated by SIRT4. So in the absence of SIRT4, there is less activity of MCC. And so that builds on this temporal model of regulation of branched-chain amino acid catabolism to drive adipogenesis. So PPAR gamma is a later regulator that pretty much regulates mature adipocytes, but at these early stages, SIRT4 seems to promote branched-chain amino acid catabolism. So if this is true, then knocking out SIRT4 or knocking out branched-chain amino acid catabolism should have an effect on PPAR gamma and its transcriptional potential. And indeed, if we knock out SIRT4, we see less PPAR gamma and less target transcriptional activity of PPAR gamma. If we overexpress SIRT4, we induce PPAR gamma. And then as we were working on this manuscript, one of the things that the reviewers asked us to do is confirm that these SIRT4-mediated phenotypes on PPAR gamma and differentiation are actually branched-chain amino acid catabolism dependent. So in the context of SIRT4 overexpression, we knock down branched-chain amino acid catabolism by knocking out MCC1. And indeed, we do find that branched-chain amino acid catabolism is required for this activity of SIRT4 to promote PPAR gamma. And so why is this necessary? Why is this kind of fuel oxidation reprogramming required for adipogenesis? So we think that adipocytes or pre-adipocytes rather require branched-chain amino acid catabolism because they're repurposing some of their other fuels. So glucose, and this has been published by other groups, it's been shown that glucose is diverted from the mitochondria and pyruvate is oxidized less, but rather glucose goes into the pentose phosphate pathway, generating NADPH, which is a critical cofactor for lipogenesis, which will allow you to store lipids in adipocytes. And then we and others have also found that glutamine is not oxidized in these cells, but rather glutamine is synthesized. And it's not really clear what the role of glutamine in adipocyte is yet. Why would these cells want to synthesize glutamine? So this is something we're also working on. But beyond these questions, we're really interested how mitochondrial dynamics are driving these choices. Is there something in the regulation of mitochondria that defines what kind of fuels they will use, and then subsequently will define these cellular outcomes and choices of how does a cell become an adipocyte or an osteoblast, and how is that all regulated by mitochondrial dynamics? So that's something that my lab is just getting into now. And so just to acknowledge people that have worked on this lab, these are my current lab members working on some of these studies, but a lot of this work was done during my postdoc in Marsha Higgis's lab. And I would specifically like to highlight Jessica Spinelli for helping with a lot of the metabolomics experiments. She now has a lab at UMass Medical Center. Elizabeth Nunn and Matt Schwasta were two undergraduate students that helped finish some of these now published studies in my own lab. And thank you for your attention. I'll take any questions in the later session.
Thank you very much, Helma, Professor Sanjang War, sorry. It's really interesting how to address or to connect the metabolisms, in this case, adipogenesis, with the mitochondrial dynamics. And this is one of the questions that I'm going to give to you, because we have not any question from the audience, not until now. So we have just 10 minutes, no more, for discussion, but I would like to give you a couple of questions. And you can answer from your side, from your thoughts, these questions that could be general, but the idea is how you think about general but relevant questions related to the mitochondrial dynamics, and also the relation between mitochondrial contact sites with other organelles like in the plasmic reticulum, but also maybe lysosomes, plasmatic membrane, and so on. So the first question is related to that, is because something that has been so very well by Dr. Leonard, related to how mitochondria has to be adapted, and with the relevance in physiology, but also in disease, the adaptation of the specific tissues and organs, depending on the molecule that could be mutated, depending the process or the pathway that could be related to mitochondria. So how the mitochondrial dynamics is adapted to the organ or the tissue, like the optic nerve, retina, optic nerve, and another kind of tissues like cardiomyocytes in hair, menstruated muscle in menstruated muscles, sorry, and also liver and so on. I mean, this is a general question, but I think we have to address this kind of thoughts in the future if we want to understand very well the physiology of mitochondrial network, but also the pathophysiology of the diseases and the specific diseases we are treating and looking for new therapeutics. So if you have any comment about that, please take your hands up and the comment that you want to do about that. Maybe
so
guys,
I don't know exactly if I can give you an answer concerning how mitochondrial dynamics is regulated on each site of the lamina cribosa in the optic nerve, but I think that there is a link between the substrates for energetic substrates in the optic nerve, which is different from the substrates which are available in the eye. I guess that in the eye, it's much more glucose which is available, whereas in the optic nerve, I guess it is lactate which is available. And based on this, Obviously, there might be some pathways which are controlling the activity of OPA1, mitofusin, and DRP1, eventually differently in the myelinated versus the non-myelinated part of the optic nerve. So I have no clues, precise clues concerning this, but I guess that according to Orion Shiri High, that you may know, there is a strong link between the substrates which are available for mitochondria and the structure of mitochondria. So this might be an answer, although we don't have a clear answer right now on each side. What I just want to point is that it's probably the only nerve and only neuronal cell which is facing this double condition in our organism, which is completely physiological for optic nerve, but which is not encountered in any other nerve of the body. Either they are myelinated or they are not myelinated, but the optic nerve is the only nerve which is both myelinated and non-myelinated, with two different status. And probably there is quite a difference in the available of metabolites.
Thank you. I mean, really very interesting, your comments. So could be related to the redox status in the way you are talking about lactate and glucose, the reduced oxidative status, and then it's in a metabolic way how the mitochondrial network could be adapted and then physiologically working in one way or in the opposite way. What do you think, Professor Simmel, Thomas, about the redox status in between this? I mean, in the case you talk about is not just mitochondrial dynamic, but also the membrane contact with the endoplasmic reticulum.
Well, I think this is very important in the CNS. We had a small paper very recently where we checked for expression of ERO1, this luminal ROS producer, in multiple sclerosis. And in the advanced stage, it's the astrocytes which maintain expression of ERO1, which normally is ubiquitous, but not anymore in the MS scenario. So the idea is that this would change the availability of lactate for the neurons in that case. So I think this is, of course, it's a novel discovery, and we're working on the continuation right now, but these redox-based mechanisms definitely are expected to play a very important role. Now, maybe just very briefly at the end, it is not just, say, the abundance and even activity of these ROS producers, which is critical. It is also the release of ROS, which can be controlled. So I don't know if any one of you had seen recently, but by measuring ROS on the surface of mitochondria, it had been published earlier this year that these are only detected if there is a clear dysfunction of mitochondria, and otherwise there are actually none. So I think because of these relatively unexpected findings that were published this year, more work is still needed to fully understand this.
May I just provide you one thought? I found quite a lot of similarities between Wolfram syndrome and Walcott-Robinson dysplasia. I don't know if you are aware of that or not, but you were speaking about the distance between mitochondria and the ER, calcium, ROS, and so on and so on. And there is a paper, a recently published paper concerning the treatment of Wolfram syndrome based on an agonist of the sigma-1 receptor. I don't know if you know this story or not, but maybe you could eventually try this agonist of sigma-1 receptor to see if it brings any improvement in your cell conditions.
Yes, I was aware of this paper, and yes, I think there is a connection there. You are correct.
Sigma-1 receptor agonists are in clinical trials in Anavex.
So it's multiple years of showing that these small molecules actually beneficial for Alzheimer's disease.
For what?
Patients. For patients with
Alzheimer's.
Alzheimer's disease.
Thank you. I mean, my next question was to you, to Eugenia, is related to, based on these comments and your talk related to neuroprotection drugs, from your point of view, which is one of the couple of major targets addressing therapeutics based in the knowledge of mitochondrial dynamics and also the connection between mitochondria and the other organelles. What do you envision to the future, to the present now and to the next?
Yeah, so hopefully our small molecules will be envisioned in the clinic by 2026. So the whole lab is working very hard on that as we speak. So we have really big drug discovery program, which at that point doesn't even have anything to do with academic development. It has very different aspect, which is new to academicians, to look at small molecule drug discovery, right? So you have to develop assay for target engagement, for efficacy, for potency, for toxicity. And that's what we're focusing on. And then structure activity relationship modifications by medicinal chemists. So we have the group of advisors from biotech through this Blueprint Therapeutics network that is helping us to get the compound that will not kill anybody, right? So complex 1 inhibition is very counterintuitive approach, but apparently everything converges on this mild stress response, right? So right now, and we wrote on that extensively, not only us, the therapies that work amazingly are exercise, right? So modification of diet and exercise is now implemented to aging population across the Europe through finger studies for Alzheimer's patients. In Europe, finger and pointer in the United States. So this is this very strong emphasis on exercise, on caloric restriction, of proper diets with food. Why is that? That's all converges on energy supply, on mitochondria function, and apparently it could be, so right now exercise is the best strategy for multiple disorders. And how does it work? It actually, what doesn't kill you makes you stronger through mitochondria biogenesis and turnover primarily. So I think it's promising. I'm optimistic. I'm cautiously optimistic, but I think it's a good time right now to start looking at mitochondria targeted therapeutics for different disorders. And I had this question for both Dr. Simon, and Dr. Lenners, because when people started to develop compounds that specifically, for example, inhibit GRP1, I was terrified because that's very dangerous, I think, without clear understanding of the problems with mitochondria dynamic, to start inhibiting mitochondria dynamics, right? We do not know in what compartment even of the cell what is happening. And now we're going to propose to inhibit mitochondria dynamics through interaction with specific mitochondria proteins. So I don't think this is a viable strategy. There is no biomarkers. The complexity is such. We saw today how mitochondria are different in the same. So if you remember my axonal trafficking movie, it's even the same compartment, and mitochondria behave so differently, and they shape and probably even energetic state and everything differ dramatically. So beautiful work today emphasizing the complexity and importance of mitochondria dynamics. So I guess my questions again are, do you think that in mitochondria disorders, enhancing mitochondria turnover, biogenesis, and mitophagy would also by default engage multiple mechanisms restoring, for example, synthesis of fission fusion proteins involved in this machinery? Is it a viable strategy to multiple diseases where mitochondria could be affected?
Thomas, Gary?
Yeah, I personally think it is not a good idea. I think in my opinion, in all of these diseases, it's the metabolic setting or the ability of the mitochondria which is not correct. But the fission and fusion I think in most cases, unless it is a direct genetic disease, it's just a reaction of the cell or the mitochondrion to these changes. So inhibiting fission, in my opinion, it doesn't sound right, but I'll let Guy answer this.
I definitely agree with that. You might, try to solve one disease, but you will push people in blindness. So I'm not sure it's really very efficient. We performed quite a lot of metabolomic studies on OPA1, DRP1, MFN1, MFN2 cell lines, MEF lines, and so on, so on. So we definitely saw that there is huge consequences on the metabolism, the cell metabolism, if you affect mitochondrial dynamics. And I don't know, I think it's much better to work on these metabolomic pathways that we evidence associated to OPA1 deficiencies and DRP1 deficiencies and MFN2 deficiencies than just playing on these actors like MFN1 and 2. If you overproduce them or if you restrict them, then you definitely will go to diseases.
I agree also. So you are talking about metabolism and then moving the last, we are late, but if you want, we can ask and have a last question directed to Santi, Alma, and Julie. They are more close working on metabolism from different ways. So what do you think about the relevance of metabolism in the mitochondrial dynamics, moving in different ways, fusion, fission, and the interaction with the other organelles? I mean, the metabolism status and how to work in that way in the way to looking for new therapies. What is your general thought about that?
Yeah, I kind of like that last thought about targeting metabolism rather than dynamics. And I think in a way, by targeting metabolism, you will also feed back into dynamics, especially with the talk we saw on cardiolipin. I think those mutants, you can probably even rescue those by modulating metabolism. So you can probably reshape mitochondria because you will need a lot of these metabolites for mitochondrial dynamics, for cardiolipin synthesis. So I do think that's maybe an angle that we should think about more.
Yeah, definitely
Santi,
true.
do you want to comment something? Santi?
Oh, sorry. I was just going to say, yeah, it makes, like Elma's rationale that, targeting metabolomics actually feeds back into the other loopholes in mitochondrial dynamics, for example. And certainly in Barth syndrome, we use arginine and it helps in modulation of the phenotype. So it has worked very well with our Barth syndrome patients because it prevents muscle breakdown overnight. And we give cornstarch. So that is affecting the metabolism. And we, in fact, considered that in cardiolipin. But our patients all passed away within infancy, so we didn't get the opportunity to use it. But I think it's a very good analogy that Elma's provided. Thanks.
I have a question for you. Have you seen any? Maybe I missed this information in your talk, but have you seen any change in mitochondrial structure when you were inhibiting a CRLS1 expression, or when you have mutation in this gene?
So the crystal morphology was abnormal, but its structure-wise,
network. About the network.
Yeah, the network. No. So what other
no,
studies perform?
Because one of my French colleagues, Tim Y, published recently the fact that he could rescue OPA1 mutation by playing with the PGS1, which is just downstream of CRLS1 gene. Yeah. So I was wondering if you find any connection between the cardiolipin metabolism and the structure of the mitochondrial network.
No, so I do know that Alexandra Filipovska, who's done a lot of work on CRLS1 as well, is doing further functional work studies on our fibroblasts to see linkage with other associated genes. But she did demonstrate through studies on gene expression levels were abnormal. And it also affected the endoplasmic reticulum stress response. I didn't put it in my slides because I didn't have enough time. I crossed over 15 minutes, I think, but yeah. There was a link between a CRLS1 defect and endoplasmic reticulum stress response as well. which is published in your paper here.
Any comment, Yuri, from your side, from your experience working with translocation and mitochondrial signaling?
So for translocation is actually its connection to the metabolism and signaling is very little studied. For example, even the contact site role is not so much studied. There was a recent paper that showed that actually, so the problem with the targeting is that it's now clear, more and more clear, that the precursors are, the precursor protein are important players in proteostasis, and cells really try to alleviate the influence. And if they happen to be in cytosol, they start accumulating and precipitating other proteins with them. So it's very important to handle them. So one of the pathways is for precursors to bind the ER surface, and then they get handed over to mitochondria. So ER surface itself can chaperone the proteins. But then it's not really clear. They think that the contact site is really important, but the role of the contact site is not studied. And also in the ultrastructure perspective, usually contact site in the inner membrane is opposing some specific subdomains of mitochondria, like it was shown for CoQ symptom that is there. But for example, what is the role in the mitochondrial ribosome and protein import? Is it somehow coordinated? Protein translation from the inside and delivery from the outside? It's not known. Yes.
Okay, thank you so much. You have a question, comment? Maybe the last one?
Yes, a very quick question to Yuri. Is this last mechanism that you talked about where TOM70 is active? Because you conspicuously did not say anything about TOM70 in this import pathway.
Yes, this pathway that I just mentioned is called ER-SURF, and TOM70 is involved in it, yes. And also TOM70 has a major role in between mitochondrial import and cytosolic chaperone system.
Okay, thank you. We have to finish because we are late. I mean, we have some comments from the journal. I mean, Bella, do you want to make some comments from the journal of Translational Genetics and Genomics for all of us and the audience?
Yes, thank you so much. And I will give a brief introduction for our journal. Okay.
Okay, thank you so much.
Yeah, thank you. Nice to hear your wonderful presentation and wonderful discussion. And I hope this conference can provide you with a good platform for academic exchange. And today, as the organizer of this webinar, I would like to make a brief introduction of our journal on behalf of our editorial office. Yes.
Okay, thank you so much. Thank you all of you, also the attendees to the meeting. And Bella, you are going to show this presentation just to finish.
Okay. This part is about our brief introduction of our journal. And the Journal of Translational Genetics and Genomics is launched by December 2017, and it is an international peer-reviewed and open access online academic journal. And currently, our journal has been indexed in the databases of Scopus, Google Scholars, NKI, and Dimensions. Yeah, and then next part is about our citation. It is very pleased to let you know that our journal articles has been cited 291, and the fact that our achieved such a good results proves that our journal has a promising future. And now our journal is to promote the translation of basic research into clinical practice. The aims of the journal are to publish high quality and original peer-reviewed research articles of discovery in the area of genetics and genomics. And so far, we have published 126 articles and 27 special issues and organizing 3 webinars. Okay, and this part is about our editorial team. Our editorial team is very international and professional, including 16 high-quality experts from 10 countries. They all play a very important role in the development of our journal. This is our editor-in-chief, Professor Sanjay Gupta. He's from Case Western Reserve University, and he is our honorary editor-in-chief, Li Jin. He's from China Fudan University. And next part is about some innovation activities of our journal, such as webinars and special interviews. And through the webinar and the interviews, we would like to bring more forward-looking voice to relative field. Our journal will continue to release more special interviews series and webinars to build a multi-channel communication platform for more scholars in genetics. And this part is about our media platforms. We will promote our latest articles and the latest activities in the media platform. You are welcome to interact with us. Yeah, this part is about our databases such as OneFoundDate and Google Scholars in KI Dimensions, ResearchGate, and Scopus. And this part is about some advantages of our journal. The first is articles are freely accessible online forever and free of charge and repeat publication. The copyright of articles belongs to authors, and opportunity for webinars and special interviews. And the last is free promotion services. Okay, and if you have any question for us, please be free to contact us. Yes, this is our brief introduction of our journal. Thank you very much for your hearing and attendance in our webinar. I hope you will have a most enjoyable conference experience here. Please be free to contact us if you have any question. Thank you so much.
Thank you so much, Bella, and thank you all of you. If there's any other question, or if not, we can finish the webinar. Has been a very nice, short discussion, but interesting discussion. Different approaches to mitochondrial disease and how mitochondrial dynamics is involved in biology and also in disease physiopathology. And thank you for it. I hope to see you in another opportunity, and goodbye. Okay.
Bye. Thank you.
Bye. Thank
Bye.
you.
Thank you.
Bye.
Muchas gracias. Adiós.
Adios. Bye. Bonjour.
Thank you so much.